So as we were saying, we're shifting away from just focusing on what's happening inside the cancer cell, because we've just been talking about up until now the cancer cell just kind of almost functioning in isolation. But we know that's not true, that there must be a back and forth between the cancer cell and the surrounding tissue, the stroma. And so that's what we mean when we're talking about this idea of back and forth communication, a dialogue instead of just being a monologue. So the question is, what role does this tumor stroma actually play in cancer progression? So as a reminder, here we have epithelial cells. Remember, the epithelial cells sit upon a basement membrane and they detect that basement membrane. That basement membrane separates them from the stromal cells or the connective tissue underneath. And all kinds of messages, molecular messages, like growth factors, things like that, move through this basement membrane. Here on the right is a nice colorful image. Again, here is some sort of a epithelial cell lined duct. This might be some sort of a glandular duct, or it may actually be, for example, the colonic crypts that we were looking at. And we're looking straight down. And so these would be the walls of the crypt with the epithelial cells. The epithelial cell nuclei are stained in dark blue. And then you see the bright green are different types of adhesion proteins that you can see are in between the epithelial cells. In this case, these are E-cadherins that allow the epithelial cells to sense that they are touching neighboring epithelial cells as part of encouraging them to stay differentiated. The bright red stain is the basement membrane. And so you can see how these epithelial cells nicely line up and they all contact the basement membrane. So what is the basement membrane? In the previous images, it kind of looks like the basement membrane is some sort of like an actual, almost like a cloth or something like that. It's important to keep in mind that the basement membrane is really just a specialized form of what we term the extracellular matrix. So the ECM is technically any of the protein environments surrounding the cells in our body, not just epithelial cells, but any of the cells in our body, of course, are going to have some sort of proteinous substance around them, right? And that's the ECM. So the basement membrane is a highly kind of modified version of the ECM that really only applies to things like epithelial cells. So you can see here in this cartoon, we are pretending that we're zoomed in and looking at the surface of a cell. So this dark gray layer would be the cell membrane. Below this would be the inside of the cell. And so above it, just outside, would be this special extracellular matrix that we call the basement membrane. And it's made of a combination of many long fibrous types of proteins. In red here are collagen, in blue are laminin. And you can see this is why we kind of think of the basement membrane almost like a true membrane, and why it looks like almost like a cloth in the previous slide, the electron microscope image. So really, it's just a pretty dense sort of meshwork of these protein fibers. And you can see these little green things that are poking up out of the cell are the integrins, right? And so these integrins, remember, they are made up of two chains that specify what they can detect or bind to on the outside of the cell. And in this case, this integrin's bound to this laminin here. Maybe this integrin that's picking up detects and binds collagen fibers. And once it binds the basement membrane, right, that encourages the shape of these integrins to be altered so that on the inside of the cell, the part of the integrin that sticks in the cytoplasm, the shape will reflect the fact that it's detecting the basement membrane proteins out here, and that would encourage then signals to be sent to the nucleus of the cell, allowing this epithelial cell to know that it's properly attached to the basement membrane, and therefore it should, for example, focus on being a fully differentiated cell and not worry about things necessarily like going into senescence or taking a break into G0. So the basement membrane provides two things. It provides anchorage for the epithelial cell, and importantly, it provides storage of all the mitogenic signals that are arriving. So if you imagine up here somewhere might be the stromal cells that are releasing different types of growth factors. In order for those growth factors to arrive at the surface of the epithelial cell, they're going to have to pass through this basement membrane. And this basement membrane, because of its mesh-like properties, slows down or captures a lot of these growth signals, dampening these mitogenic signals from reaching the epithelial cell. So if there's going to be lots of interactions between the stroma and things like cancer cells that are really becoming disease-like, do you really imagine that the stroma is going to look the same for all different types of cancers? And the answer is no. Even within a particular type of cancer, the stroma within a tumor can look dramatically different. Here are two examples of breast cancer. One form here we call ductal carcinoma, one form we call lobular carcinoma. Let's look at the left first. In ductal, the brown stained cells are the epithelial cancer cells, and the light blue stained cells are the stroma. So keep in mind, this is a cross-section of a large tumor, right? So this whole mass would be considered cancerous, and it's made up of epithelial cells and their stroma. So you can see there's quite a bit of stromal cells here, right? And the stromal cells seem kind of spaced apart, not very dense. Compare that to the relationship that you see in lobular carcinoma. Here, the navy blue stained cells are the epithelial cancer cells, and the really dark black stained cells are the stroma. And you can see they're very different. They're really compressed together, much more dense, not nearly the same amount of stroma in between. So this implies then that the stroma itself must be changing as a result of the epithelial cells becoming more and more diseased. So what is stroma? We already talked about that the stroma is made up of long protein fibers like collagen, fibrins, laminins. But of course, there's a whole population, a mixed population of different types of cells that make up the stroma. This is a list of sort of what's considered the 9 different types of cells that you can expect to find as part of stroma. So let's look on the left here, another example of tissue. Here you have in purple the epithelial cells that are lining various ducts, and then all of the orangey, sort of pinkish cells would be considered a mix of these various types of cells that make the stroma. So these can be broken into ones that supply structure. So these types of cells here in the stroma provide structural support. They actually allow the physical support for the epithelial cells to form structures like these ducts. Then you'll have some cells that are vascular in nature. So vascular means having to do with our circulatory system and blood supply. Because clearly throughout this tissue, there must be a pretty extensive network of things like capillaries, bringing nutrients and oxygen to supply these epithelial cells and allow them to grow. Then we have immune cells. We oftentimes don't think about these, but throughout the stroma, you will have a lot of immune cells that are patrolling and helping to regulate, either detecting things like disease cells, or even just managing and getting rid of things like older, healthy cells that just need to be recycled. So this whole mix then of different cells in the stroma, you can imagine can be a very confusing place because each of these types of cells has their function. So therefore, we have what's called heterotypic signaling, simply meaning you have lots of different kinds of signals going between many different types of cells. And one way to think about this is that each cell type within the stroma is sending messages that either encourage or limit the proliferation or the activities of the other cell types that are all mixed in there. And together, that balance of heterotypic signaling might vary from tissue to tissue. So this is a nice reminder also that in our class, we focus on epithelial cell cancers, so carcinomas as a model for understanding cancer. And so our discussion today about the relationship between epithelial cells and stroma might be more specific to carcinomas, but many of the themes that we're going to talk about still apply to other types of cancers and different types of tumors as well. So how important is it that the tumor cells, the epithelial tumor cells, still have correct messages coming from stroma? This is a cartoon from the book that depicts some early experiments that were done, human experiments that were done, I think, in the 1950s. And these experiments seem very remarkable today because back then there was very little oversight in terms of what was considered sort of ethical or things to try. But what they did was they would take, in patients who had, let's say, a tumor, let's say in the arm, they would take some of the cells from that tumor and actually try to transplant them somewhere else on the patient's own body, for example, on the back over here. And what they found was that if you just took the epithelial tumor cells alone and transplanted those, these cells, once they're transplanted, let's say from the arm to the back, they would struggle to grow. So in the images down here, on the right, you see an example of what these transplanted cells might look like after some time if you only took the epithelial cells and none of the stroma that were part of the tumor. And these cells, you can see, are very, they don't look very happy. They're not growing very well. In fact, they look like they're kind of senescing. However, when these researchers took a little bit of the stromal tissue from the patient's tumor and transplanted that along with the epithelial cells, then they could actually see that the epithelial cells would start organizing and forming a new tumor in the new location. So again, this is very remarkable. I'm sure some of you are raising your eyebrows, and it's hard to believe that back in the 50s or so, they could actually do these types of experiments on people, right? So clearly this is something that we would not do these days, but at least we can benefit from this knowledge that was gained. So processes that we associate with tumors becoming large and aggressive and succeeding in metastasizing or moving out of their original tissue location, they depend upon the stroma in the new location actually becoming modified and helping out the new cells, the cancer cells that have moved there. If we can see that when we look at the impact that metastatic cancer cells have when they move from one organ where they started and arrive at a different organ in the body. You can see here, these are metastases of different types of cancer that have both arrived in the liver. So in this case, we're saying the original cells were, for example, colon cancer cells that metastasize from the colon and now have arrived in the liver, or these are breast cancer cells that originated in the breast tissue, metastasized, but have also arrived in the liver. And we can see that the impact they have on the liver tissue is dramatically different. Here, the colon cells that are stained in blue, so these are the metastatic, fully transformed cancerous colon cancer cells, right? Now they're in the liver. Over here, you can see this section of the liver is still considered normal. So this is what the normal liver stroma might look like. But as you get closer to where the metastatic epithelial tumor is growing, you can see the tumor begins to change. It looks very different. And in fact, it's changing in such a way that now it's sending messages to the tumor cells that are actually supporting and encouraging these colon tumor cells to form things like ducts, right? They form structures as though they are like forming the new colonic crypts that used to be in the intestines, but now they're very much out of place, and they're forming these crypts in the liver with the support of these changing stroma cells. On the other hand, if we look at the case of metastatic breast cancer, Again, these dark cells over here, right, would be the metastatic breast epithelial cells that have arrived in the liver. And you can see here, this would be the normal liver stroma. And what you begin to see is that the stroma in this case is much more passive. It is being shoved aside as the metastatic epithelial cells sort of invade the stroma. And unfortunately, the stroma, so this part on the rightmost side, you can see is a little bit different. That would be the normal liver stroma again. But as you get closer to where the metastatic cancer cells are moving over, you can see the stroma itself is changing. And in this case, the stromal cells are getting out of the way to allow the epithelial cells more access to the blood supply. These little red dots that you can see scattered over here, these are all cross sections of capillaries, because these are all little red blood cells that are flowing through. So this implies then that there's this back and forth communication between epithelial cells and stroma. And the stroma might behave very differently depending upon what type of cancer is invading it. So that's why we can think about this as sort of this analogy of a rebelling teenager. So we have a household here where, you know, it used to be that it was really nice and the family lived very sort of harmoniously under one roof. And that's because when the teenager, when she was a little girl, maybe all she did was she listened to her parents and she behaved really well and followed their instructions, just like a normal healthy epithelial cell would. But as she becomes a teenager, just like as the normal healthy cell begins to transform, right, she gets very kind of rebellious, right? As the normal epithelial cell gets more and more mutations, it begins to no longer listen properly to the types of messages that were originally coming from the stroma. So the important thing here is that even though we have the rebelling teenager and she's making it very difficult and she's causing a lot of confusion and even manipulating her parents, right? The whole point is, at this point, she's still not ready to be able to live outside of the house. In other words, at this point, the transformed epithelial cell still depends upon the original stroma to support that cell. And that's why we say originally, as the tumor starts to form, it is still contained in situ, right? In situ is a term that means in the original site where the tissue originated from. And you can imagine then that this cartoon of the house sort of containing this family that's becoming more and more dysfunctional. One way to think about it is the walls and the roof of this house can kind of represent, figuratively speaking, the basement membrane, right? And the rebel teenager still has to sort of survive within the supporting framework of the stroma and the basement membrane.